Bismillahirrahmanirrahim. My name is Mubashir Wahab and I'm the student of M.Phil microbiology second semester, and the topic I have to present is antibiotic resistance, and this topic is being presented to Dr. Niaz Muhammad, and this topic is being chosen from the course contents of recent trends in molecular biology. So first of all, here we have to know what is antibiotic resistance. So just by the name we can observe its meaning, like antibiotic resistance against the antibiotics. It is the resistance against the antibiotics being developed by the microorganisms due to the repetitive use of antibiotics, making it harder for the antibiotics to treat an infection caused by the bacteria or caused by the resistant microorganism. So we have the example of methicillin resistant staphylococcus aureus, which is also known as MRSA. It is a bacteria that have the resistance, that got the resistance against the most commonly used antibiotics in our society, most commonly being prescribed by the doctors, making it harder for an infection to be treated. Like in the first plate, as you can observe, we have placed many antibiotic discs, and you can see the colonies of bacteria are being sensitive to these antibiotic discs, and we can observe many zones of inhibition. But in the second plate, as you can see, we have only few zones of inhibitions, and the rest of antibiotic discs, they are not faulty. It's actually the bacteria that got the resistance against them. The antibiotic discs are working perfectly, but the bacteria have the resistance against them. That's why there are no more zones of inhibitions, and the bacteria is no more sensitive to these antibiotic discs. So that's what being problematic for us, because we cannot treat the infection anymore if the antibiotics are not working on these bacteria. So moving toward the next slide, we have the mechanism. Like now we know what is antibiotic resistance, but we have to know what is the mechanism, how they develop this antibiotic resistance against the antibiotics. So there are 5 mechanisms of antibiotic resistance. As we can see, the first one is antibiotic inactivation. Just by the name, as you can see, the bacteria have the ability to inactivate these antibiotics just by releasing some enzymes, as we have the example of beta-lactamase. Beta-lactamase, this is an enzyme that is being released by the bacteria against the penicillin, because penicillin have the beta-lactam rings in its structure, and when its structure is being degraded, it's no more working on a bacteria. So the beta-lactamase plays a role of degrading the structure of beta-lactam ring in a penicillin, or also the cephalosporin. So once these microorganisms degrade these beta-lactam rings, these antibiotics are no more working. This is the first strategy that the microorganisms play against these antibiotics. Now the second is prevention of antibiotics entry into the cell by reducing the permeability. So what they do, the microorganisms, they modify their membrane proteins, which are also known as porins, which are used for the diffusing in and diffusing out of the different molecules, including antibiotics also as well. So what they do, they would just modify these porin proteins, making them more narrower, so that the antibiotics can diffuse inside the microorganism and cannot play its role. So we have the example of Pseudomonas aeruginosa, which has the resistance against the imipenem. What they do, they would just modify their porin proteins. Now in the diagram here, you can see, we can observe different strategies being followed by this microorganism. This whole structure is of a bacteria, and the first one, as you can see here, antibiotics, when they would just enter inside the bacterial cell, and you can see this structure is being degraded by these these enzymes. What these enzymes will do, they will just degrade this structure, and this structure is no more functioning like its original shape. So it's no more working like its original form. It's no more in its original form, so it cannot perform its activity. It cannot degrade the microorganism. It cannot perform its activity. So it is degraded by these enzymes being released by this extra chromosomal DNA virulent gene is being stored in the extrachromosomal DNA, which is also known as plasmid. Plasmid plays a main role in this all activity. Now the second role, as you can see, we have the antibiotics altering the enzymes. We will study this topic in the next slide, but I'm just trying to represent it here. As you can see, antibiotics, they would just enter inside the cell. What these enzymes would do, they would just alter the shape of the antibiotics, or they would just start to bind these antibiotics, and these antibiotics will no more be able to work because all their binding sites have been occupied. They cannot bind to any other structure. They cannot degrade the microorganism. And now here in the antibiotics, as you can see, we have the main role being played here is by the efflux pump. What these antibiotics, they get their possibility to enter inside the microorganisms. Like here, as you can see, they are inside the microorganisms, but as they enter the microorganism, they are immediately pumped out of the cells. As they would enter, they will just be pumped out of the cell, and they cannot perform their action. So this function is being played by the efflux pump, and this gene is being stored inside the extrachromosomal DNA, which is also known as plasmid. So as you can see, these arrows here, and this one, all these genes are being integrated into the plasmid, and this virulent factor is being played by the plasmid. Now we have the next slide of mechanism of antibiotic resistance. We have the third one, active efflux. Like I just explained, active efflux of antibiotics, it is the active drug export of drugs. Like the antibiotics, we would just try to ingest against an infection. So when we ingest these antibiotics, what the bacteria would do, these antibiotics would enter the bacterial cell, and they would just immediately be pumped out of the cell actively. And we can see this in Enterobacteriaceae family. The example of this mechanism being followed by this family to show the resistance against these against these antibiotics. Now the fourth one we have is the modified targets. What the bacteria would do, they would just modify these targets to which the antibiotic have to bind. For an instance, bacteria have a structure to which the antibiotic have to bind to degrade the bacterial cell. So what the bacteria would do in response, they would just modify these targets so that they cannot be recognized by the antibiotics anymore. For an instance, we have Streptococcus pneumonia having the modified penicillin binding proteins PBP. So these, after modifying these target sites, the antibiotics are no more working, showing that the microorganism is now resistant to these antibiotics. Now the fifth one we have is bypassing drugs action. So what the microorganism would do, they would just release the modified targets. They would release the modified targets along with the normal targets too. They won't stop the normal protein binding sites, but they would also release the alternative protein binding site PBP, which is also an example shown by the MRSA, methicillin resistant Staphylococcus aureus. They would release these alternative sites to which the antibiotics would bind, and these normal sites would then be safe. So this is how they show the resistance. The bacteria is not killed, it is not dead, it still continues to increase their number and cause infection. Now moving towards the next slide, we have two different cells. The first one is non-resistant cell, and the second one is resistant bacterial cell. And the first one, as you can see, the drug would get the entry. The drug gets the entry here, and this is the target to which it binds, and it would cause its action and destroying the cell. But as you can see in the second one, the drug would get the entry, which is a resistant cell. So what the cell would do, they would just be actively pumped out by the efflux pump. So these antibiotics are no more working on it. We have the inactivating enzymes. When these antibiotics would just enter inside the cell, these inactivating enzymes would bind to it, making the antibiotics unable to bind to any structures because their binding site has already been occupied by these enzymes. The third one is modifying their foreign proteins. So what happens here is when the antibiotics would just try to enter the bacterial cell, they would just not get their entry because the foreign proteins are being modified now. They are more narrower. They won't allow the entry of antibiotics. So what happens here? These antibiotics are being repelled. They don't get the entry, so these antibiotics are no more working on it. And here we have the fourth one, in which we have the alternative targets. When the antibiotics would get the entry, their structure is different, and their target is now different. So what happens here? These antibiotics don't work on it, and it's no more able to perform its action. So bacteria can't perform any infection. Now the next classification of the resistance we have is higher level of resistance and lower level of resistance. High level of resistance refers to the resistance that cannot be overcome by increasing the dose of antibiotics, because it is being mediated by the enzymes, by the production of enzymes, such as we have the beta-lactamase enzyme that is being released by the microorganisms as a response against the antibiotics. So what is the function of beta-lactamase ring? They would just degrade the structure of penicillin. They would just degrade the beta-lactam ring inside the penicillin and cephalosporin, so that these antibiotics are no more working against these microorganisms, showing the resistance. Now we have the lower level of resistance. It refers to the resistance that can be overcome by increasing the dose of antibiotics, because this kind of resistance is being mediated by the mutation in genes. So once the genes are mutated, the structures they would just produce will be also different. But still, even that these structures are different, still the antibiotics can still bind to it, because some of its portion are still matchable. So the antibiotics can still bind to these structures and reduce the resistance, and can overcome the resistance. Moving towards the next slide, we have origin of antibiotics resistance. We have the genetic antibiotic resistance and genetic basis of antibiotic resistance. Now first we have that there may be accumulation of bacteria that the concentration don't allow the antibiotics to pass through them, so therefore the antibiotics are no more working. Surgical drainage is therefore necessary. Second one is that bacteria can be in a resting state, they are not growing in numbers, so therefore they are not detectable to these antibiotics that have the activity against the cell wall. Now the third one is that when under certain conditions, when the penicillin acts upon a microorganism but not totally degrading them, just degrading their cell wall, but still they survive as a protoplast. What is the condition? That these antibiotics are no more working. If we just ingest the antibiotics because of an infection, so this cannot work on these microorganisms because they don't have their cell wall anymore. So these antibiotics don't have their activity on these antibiotics. Some foreign particles, if they are present there, they are making it difficult for the antibiotics to work against the microorganisms. Now there are some artifacts that play an important role in making it look like that these microorganisms are resistant to these antibiotics. The first one is that if we take wrong drugs, obviously if we have a different infection and we are taking different antibiotics not specific for that infection, so obviously they won't work. The second one is if you take the wrong dose. For example, if it would require 500 mg of these drugs and we are just taking 50 mg, obviously it won't work. Or if we take the outdated drugs, obviously it won't work. So what is the genetic basis of antibiotic resistance? Genetic resistance can also occur through the extrachromosomal genetic elements named as plasmid. Like I explained before, every virulent gene is being stored in the plasmid, which is the extrachromosomal DNA. So if a bacteria having a resistant plasmid, obviously they would show resistance against these antibiotics. So first of all, we have to know how they get this resistant plasmid. There are three main ways. First one is transduction, conjugation, and transformation. Moving towards the next slide. So what is the transduction? Transduction is the exchange of genetic material between two bacteria from donor to recipient through the viruses known as bacteriophages. These are the specific viruses for the bacteria. They only attack bacteria, and they use their cellular machinery to synthesize their own structures as well in increasing numbers. So what they do when a virus attacks donor bacteria, acquiring its genetic portion of DNA and integrating it into its own by an error, and later on when it attacks the recipient bacteria, transferring this genetic portion acquired from the donor bacteria and increasing and replicating it into many numbers, helps the bacteria to acquire the resistance. Now, what is conjugation? Conjugation is the process by which the bacterium transfers genetic material to another bacteria through the direct contact. During conjugation, one bacteria serves as the donor of the genetic material and the other serves as the recipient. The donor bacteria carries a DNA sequence called the fertility factor or the F factor, which is being transferred to the another bacteria through a sex pili, through a bridge that is being developed between the two bacteria. So the one bacteria, the donor bacteria, being resistant and the recipient bacteria being non-resistant, and after the transfer of these genetic material, both bacteria have now the resistance against the antibiotics. Now, what is transformation? Transformation is the process by which the foreign DNA is introduced into a cell. Now, the other bacteria that would just die in the environment, releasing their internal contents out, including their genome, including their DNA. So this DNA is then taken up by the living bacteria that comes in contact with that genome part. Now, integrating this external DNA into their own, they may get the resistance against some antibiotics. This external DNA can enhance their characteristics and potentially introducing a new traits like antibiotic resistance. Now, moving towards the next slide, the misuse of antibiotics selects for the resistant mutants. Now, what are the misuses? They are the using of outdated, weakened antibiotics. If you use the outdated and weakened antibiotics, obviously they won't work against the microorganisms, but in return the microorganisms will get the resistance against the working ones. So the using of antibiotics for the common cold and other inappropriate condition. If we would just use antibiotics for every sick we feel, every time we got sick, we will just ingest these antibiotics. Obviously, these microorganisms that are not responsible for causing that illnesses still will get this resistance against these antibiotics, because we are commonly, repeatedly using these antibiotics. And the use of antibiotics in animal feed to promote the growth. If you would just mix the antibiotics with the feed of animals to let them grow, to promote their growth, but obviously the microorganism in them will just get the antibiotic resistance. Obviously, failure to complete the prescribed regimen. So if a doctor prescribes us a course of 6 days or 7 days, and we just after completing 2 days of course, and then we just stop using these antibiotics anymore because we just feel fine, but it's not, because there are resistant bacteria left behind and they would just grow in number, and for the next time these antibiotics will not work against them. The bacteria would just get the resistance. It would just use someone else's leftover prescription, and some physician prescribe multiple drugs when one would be sufficient. Obviously, they would just give you the combination of different antibiotics when only one type of antibiotic would be sufficient for curing that disease. And in some countries like in Pakistan, in India, in Bangladesh, the antibiotics are available on different medical stores without the prescriptions. Now, moving toward the next slide. How can we slow the emergence and spread of antimicrobial resistance? How can we stop it? We have different responsibilities of physicians. They must work to identify the microbe and prescribe suitable antibiotics. They must perform different tests on patient sample, and then based on the results they have to prescribe these antibiotics. And what are the responsibilities of patients? They need to follow carefully the instructions given by the physicians. They don't have to just take the antibiotics for 2 days and left it over, while the course is of just 6 days. So they don't have to complete the course in just 2 days when the course is being prescribed for 6 days. We must educate the public, we must spread the awareness about the resistance being developed by the viruses, being developed by the microorganisms such as bacteria, fungi. We have different microorganisms that can acquire resistance. So we must educate our public, our people, so that they know they don't have to take antibiotics for every illnesses they would face. And what are the global impacts? Organisms that are resistant can travel very quickly nowadays, because humans would just travel from one country to another, from one region to another region, taking these microorganisms, resistant microorganisms, with them as well. Now, what are the new approaches of antibiotics therapy that are needed? Our scientists need to work to find new antibiotics, new targets, and new strategies to be followed. And they can also use the new combinations of different antibiotics. Discovery of a new and unique antibiotics as is necessary, so that these microorganisms can have resistance towards it, saving many lives. Thank you. So that was my presentation. Thanks to all of you for listening.